The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone, and thank you for joining us today. You have logged on to the Nelson Labs webinar, the Reprocessing Rethought on Effective Cleaning and Sterilization of Robotic Devices. We just have a few housekeeping items before we turn the time over to Griffin. But if you missed one of our webinars or would like to refer back to it, you can always find them on the Nelson Labs website on the on-demand webinars page listed under Education. You can receive notifications on upcoming live and on-demand webinars by following Nelson Labs on Facebook or LinkedIn. We also invite you for free to register for the Sotera Health Academy at soterahealth.com/academy. There you will find curated content from industry thought leaders from Nelson Labs, Sterigenics, and Nordion, and our regulatory compliance associates at a Nelson Labs company. We would also like to invite you, as a reminder, about the Nelson Labs seminar. Nelson Labs performs industry-wide seminars throughout the course of the year. And the aim of this 3-day seminar is to provide medtech professionals the opportunity to establish or refresh their fundamental testing knowledge and thereby achieve more efficient, accurate, and testing outcomes. For a 10% discount as an attendee of today's webinar, use code SEMINARS10 when registering. Today, we are joined by Griffin Kamick. Griffin is the Senior Expert Consultant for the Healthcare Reprocessing Section at Nelson Labs. He graduated from Brigham Young University, Idaho, with a bachelor's degree in biology and has been with Nelson Labs since 2014. We do encourage your questions. Submit them anytime in the questions box so we can have an open forum discussion. The Reprocessing Rethought is intended to be more conversational with audience questions filling a majority of the time, so please submit them at any time. Now I will turn the time over to Griffin to get started.
Hello, and thanks, Mike, for that introduction. As mentioned, my name is Griffin Kamick, and I am the Senior Technical Consultant at Nelson Laboratories. And today I wanted to kind of have a discussion about effective cleaning and sterilization methods for reusable medical devices, in particular, reusable robotic medical devices. And some of the challenges that may arise when trying to validate these processes for, in particular, for these robotic devices, as they tend to be a bit more complex than your average reusable medical device. I've been with Nelson Laboratories for, as Mike said, for over 10 years, primarily working in the reprocessing section. So I am very excited to have this discussion with you all today. I am very passionate about reprocessing validations. It's what I think about all the time. Even when I'm at home doing my own dishes, I'm thinking about how to effectively clean them. And so as I said, I'm excited to go over this topic, and hopefully we can have a good discussion today as we discuss these items. I just wanted to do a bit of a background just to kind of reiterate and review some items, primarily just kind of give, that the definition of a reusable medical device, just to ensure that we're all talking about the same type of devices and that we're covering the same or the correct cleaning and sterilization processes. So a reusable medical device is a device that is intended for repeated use on multiple patients with appropriate decontamination or other processes or processing between uses. So so these devices can range from your non-invasive devices like the and non-patient contacted devices like the monitors here to the more invasive devices like surgical robotic equipment, which is what we'll be kind of going over today. And again, to kind of reiterate and go over reusable medical devices can be categorized into 3 different classifications using the Spaulding Classification. This classification is really just based on how the device interacts with the patient, and it will let you know what reprocessing instructions are needed for that particular device. So for those devices that are either non-patient contacting or will contact intact skin, these are classified as non-critical devices. So these are your items like blood pressure cuffs, monitors, carts, things that have low risk associated with them, and really only need to go through cleaning, and then in most cases either low level or intermediate level disinfection. The next classification are your semi-critical reusable devices. These are devices that are intended to contact mucous membranes or non-intact skin. So these are items like speculums and endoscopes. They have a bit more risk associated with them than non-critical devices, but they're not contacting the blood pathway or sterile parts of the body, so there's still a little bit less risk associated with them than compared to other devices. And with these devices, semi-critical devices, they need to go through cleaning and then ideally sterilization. However, a lot of semi-critical devices cannot be sterilized, and in those cases, high-level disinfection may be appropriate. And high-level disinfection is the minimum requirement, essentially, for semi-critical devices. And then the last classification are the critical devices, which these are devices that are going to contact sterile parts of the body and the blood pathway. So these are your surgical instruments, such as the robotic devices that we'll be discussing today. And they have the most risk associated with them. As such, these devices need to go through adequate cleaning, like the other devices. However, the terminal step does need to be sterilization with these devices. High-level disinfection or disinfection can be an interim process, but the terminal process for critical devices does need to be sterilization. So as we have our discussion today, keep in mind that robotic surgical devices, reusable robotic surgical devices, are going to be classified as critical devices. So they're going to need to go through, again, an adequate cleaning procedure that demonstrates that, that will remove kind of all the contamination from the device, and then they need to go through sterilization at the end to ensure that the devices are sterile and safe for subsequent use. And so that's just the quick review I wanted to go over before we kind of start our discussion. And so at this time, I'll kind of turn the time over to you all to ask any questions and to kind of go over any items that you wanted to discuss or bring up, any topics kind of concerning robotic devices and kind of the challenges in validating these cleaning and sterilization procedures for these particular devices. So feel free to write your questions into the question section, and we'll get the discussion started.
Thank you, Griffin, for kicking off this discussion. We ask again, I'll just reiterate that please go ahead and submit your questions at any time. We'll use the remaining time of this session to discuss with you the things that are on your mind regarding robotic devices. So Griffin, here's the first question. How would you classify the robotic arm under a sterile cover?
That is an excellent question. So that is, and it's a little bit of a tricky question. So the robotic device that's going to be contacting the patient, as I mentioned, that will be classified as a critical device. And so it's going to go down to the central processing or the sterile processing department, and it'll be cleaned and sterilized. But the robotic arm, that's often a really large piece of equipment, but it's not patient contacting. And as mentioned in the question, oftentimes it's going to be covered, or at least is intended to be covered with some sort of sterile covering or sterile drape. And so with those parts of the device, I would classify that as a non-critical device, or that portion of the robotic equipment as non-critical. So kind of going back to the Spaulding classification that I discussed earlier, you're going to need to ensure that that part of the robotic equipment can be adequately cleaned. And in this case, I would probably recommend an intermediate level disinfection, just in the off chance that that sterile drape is compromised and maybe the robotic arm comes in contact with blood or other bodily fluids during the procedure. Now, I know in these procedures, I think they're not intended to be see a lot of blood or other contamination during these sorts of procedures, but I would still, in this case, just due to where the robotic arm is being used in reference to the patient, and as well as kind of the nature of the procedure that's happening, I would recommend an intermediate level disinfection. But that is an excellent question, because that is kind of a tricky part with these devices, as you have the robotic part with the end effector that is patient contacting, and then you have the arm higher and up above that's going to be draped that is not patient contacting. So excellent question.
Great, thanks, Griffin. Here's our next question. So what sterilization modes are most common with robotic devices?
Yeah, that is a great question and a difficult one to answer as well. And it really just depends on what the material and the mechanisms of the robotic device can withstand. If they can withstand, if the material and the mechanics of the device can withstand steam sterilization, then that'd be my first recommendation. And that's a very common modality, and one that I've seen with some other with robotic devices. And it's the one that most hospitals are going to have access to, is that moist heat or steam sterilization. But as we all know, not every material and not all devices can withstand such high temperatures and pressures, and so you may want to look at alternative methods such as vaporized hydrogen peroxide or ethylene oxide. Those again have their own limitations with material and electronic equipment as well. So really, when you're choosing your sterilization method, you really are going to have to take into account the design of your device, the materials of your device, and just the mechanics of the device as well, because you want to make sure that the device after sterilization can still function properly. But that is an excellent question as well.
Great. Okay, on to our next one. What is the difference between high level and intermediate disinfection?
Yeah, so high level disinfection, again, that's going to be for your semi-critical devices. And you are going to use a mycobacterium species to challenge the high level disinfection process. And you need to achieve a 6-log reduction with that mycobacterium species. Oftentimes, high level disinfections are performed using a high level disinfectant or chemical sterilant, and the device has to be fully immersed in that chemical disinfectant. So products like Cydex OPA or Sporox 2, these are common high-level disinfectants, and they require the device to be fully immersed. So if your device can't be fully immersed, then then high-level disinfection is not going to be a possibility for that particular device. And then if you're submitting within the US, you also want to make sure that you are using an FDA-approved high-level disinfectant. So the FDA has a list of approved liquid chemical sterilants, and high-level disinfectants. And you want to make sure you choose one from that list when you go to create your reprocessing instructions and perform your validation, your reprocessing validations. Now, for an intermediate level disinfection, for that you need to demonstrate a 6-log reduction of 4 vegetative organisms. Typically, those are going to be E. coli, S. aureus, P. aeruginosa, and K. pneumoniae. As well as you need to demonstrate a 3-log reduction of a mycobacterium species. So it's less of a requirement than high-level disinfection, where you need to demonstrate that 6-log reduction of a mycobacterium species, but it's still a higher requirement than what is needed for a low-level disinfection, which is only a 6-log reduction of those 4 vegetative organisms that I just discussed. And with intermediate level disinfections, those are often going to be carried out by using chemical disinfecting wipes or sprays. So products like Super Sanicloth, Clorox healthcare germicidal bleach wipes, Cavi wipes, Clinell universal wipes. These are all common intermediate or low-level disinfectants, and in often cases they're going to be wipes. There are chemical sprays out there that you can use, but kind of the wipe is the more common product. And then again, if you're marketing within the US, you want to make sure that you are using an EPA registered disinfectant. So just make sure when you're selecting a disinfectant that you just either look at the bottle, or you can look online and you should be able to easily find that EPA register number, either just on the chemical bottle or the manufacturer's website. Yeah, that's an excellent question. The biggest thing, again, just kind of going over high-level disinfection, is that it requires the device to be fully immersed. So kind of going back to the robotic arms, the non-patient contacting parts of the robotic device that are going to be covered with a sterile drape, you would think that you want to go for a high-level disinfection with that equipment. However, those robotic arms are just too large to immerse in a chemical disinfectant, and they can't be immersed. Just they're not designed to be immersed. And so while a high-level disinfection may be the most conservative choice for that part of the robotic device, it's not going to be feasible with the chemical disinfectants that are used for high-level disinfections. And it may not even be needed, because again, it's a non-patient contacting portion that in most cases that is going to be covered with that sterile drape. And so really that intermediate level disinfection using that wipe is going to be what I recommend, and I think an appropriate procedure to demonstrate that the device can be safely reprocessed between uses.
Great. Here's our next question, Griffin. Says, can you help understand what parts of the body is defined sterile? Wouldn't an endoscope also touch a sterile part of the body?
Yeah, that's an excellent question. So endoscopes are often they're coming in contact with mucous membranes. So like a transesophageal probe, or really type of any endoscope, while they are entering a cavity of the body, they're not considered sterile parts of the body. As you have your own your microbiome within your body, it's not a sterile environment. And so again, sterilization for these devices is what's required, or what's recommended, but the requirement is at least high-level disinfection, because it's not necessarily entering a part of the body where introducing a microorganism is going to have a drastic impact. So with your critical devices, if you were to introduce those are entering the blood pathway and entering parts of the body where you generally don't have large amounts of organism. And so you're giving, if you're introducing a contaminated device into that part of the body, you are going to give that organism a really good opportunity to spread. Whereas with endoscopes, the mucous membrane contact that they're coming in contact with, there's already microorganisms there. And so it's going to have to combat whatever microorganism that you may introduce is going to have to kind of outcompete the existing organisms there. And in some cases, we do see that happen, which is why we need to ensure that we can properly clean and disinfect products like endoscopes, because while they are not coming in contact with sterile parts of the body, they can still create opportunistic infections by introducing these microorganisms, because in a lot of these cases, the patients that are undergoing these procedures are immunocompromised, and so they do have, introducing additional microorganisms may cause these opportunistic infections. And so again, with semi-criticals, the expectation with semi-critical devices, the expectation is sterilization, but the minimum requirement is high-level disinfection, because in some cases, those semi-critical devices just cannot be sterilized, just due to the material and the mechanisms of the device.
Okay, here's our next question. What is recommended classification for surgical console or 3D glasses used on RAS systems?
Okay, so just the console itself, or the I guess you said the glasses, the headgear that person would be wearing. I would again classify that as a non-critical device. While it will be used during the surgical procedure, it is not coming in contact with the patient, and so at minimum, the device, these sorts of devices need to be adequately cleaned, and then again, I would recommend a intermediate level disinfection. So these are these devices may come in contact with blood or other bodily fluids, and so I wouldn't do a low level disinfection. I would again recommend an intermediate level disinfection for these devices. Again, these consoles and these glasses are going to be too large to immerse into a chemical disinfectant, or may not be compatible with immersion. And so using a high level disinfectant, while again may be the conservative approach and maybe what you want to do, it just may not be feasible for these types of devices, and so wiping it down with that intermediate level disinfectant is going to be the best approach. And then additionally, these devices are non-patient contacting, and so the risk associated with them is lower than a non-critical or critical device. And so that intermediate level disinfection is going to be an appropriate terminal step for these devices.
Okay, here's our next question. What impact does the new ISO 17665 2024 standard on reusable medical devices, including robotic devices? So what's the impact?
Yeah, so that's an excellent question. That is a brand new standard from last year. Updated standard, so it kind of combined all parts of ISO 17665, parts 1, 2, and 3. To a degree, it combined those into one single document. And essentially, it doesn't have necessarily a large impact, I would say, on robotic devices and just in sterilization validations in general. You're still going to need to demonstrate or perform a sterility assurance level validation, ensure that you can demonstrate the proper lethality with your sterilization process using the appropriate BIs and placing those biological indicators or BIs in the appropriate locations. You know, and then additionally, just with any moist heat sterilization validation, you are going to need to ensure or perform a dry time validation as well. And then, you know, we recommend also doing temperature profiling of the chambers. But in terms of impact specifically to robotic devices, I can't think of any large impact. It's something that I'll definitely look into, but from my understanding right now and what I've read, there's not necessarily anything major or any major change that is going to impact, you know, how robotic devices are steam sterilized or sterilized. And then there's no change than what was required before, right? No major change from what I recall, but I'll definitely look into it, and I can send a follow-up if you would like as well.
Okay, we have a couple more questions that we have, I hope, time for. It says, hello, may I know the following, normally how to evaluate if a previous efficacy validation can still be representative for a design change? Do we need to revalidate if a new product is added to an existing product family?
Yeah, that's an excellent question. And the answer to that is it depends on how extensive that design change is or how different the design of that new device is. It is acceptable to adopt new devices or a design change into a previously completed reprocessing validation, whether that's a cleaning validation or a steam or sterilization validation. You just need to assess the devices that were used for these validations, and if they present a greater challenge to the process than the new design or the new device, then you should be able to adopt the device under that previous validation. That's something that I can help with if you would like. I worked on those assessments in the past, and really I can help determine whether the design change is significant enough to necessitate an additional to necessitate additional validation work, or just some additional abbreviated testing, maybe not a full validation. If your reprocessing validations are quite old, so if your cleaning validation is old, there's no requirement to redo your cleaning validation to meet the new standard for AAMI ST98. But if your cleaning validation is old enough, I would at least entertain the thought of maybe updating your cleaning validation if you're going to adopt a new device under it, to kind of redo your cleaning validation to meet the current standards. Though that's not a requirement. It's just a recommendation just to ensure that your, you know, the cleaning validation that you're trying to adopt the new device under meets the current requirements. It just kind of makes that adoption process a little bit smoother and easier. But that's an excellent question. You can definitely follow up with me, and we can discuss you know, what kind of goes into one of those assessments.
Okay, Griffin, we have a couple questions left, so maybe we'll just go a little bit over. So here's one of them. Can you share your thoughts around the recommended standard or standards and test methods to determine the residual moisture prior on instruments after cleaning and prior to steam sterilization? Do you provide such services?
So there's not necessarily a standard or a test method out there that I'm aware of. That is something that we can do. It's not something that we commonly do. I have seen that request in the past, but it's not there's not a standard that provides an acceptance criteria. So what is an appropriate amount of moisture? Is it no moisture, or, you know, what percentage of moisture is appropriate? So that's something that you would need to define. prior to testing or after testing, once when you see once we have results. But yeah, there's no standard out there that I'm aware of, but it is something that we can help with, and we can help design a test method to determine what the residual moisture is. And then we would need to also just work together to determine what what the acceptance criteria is for that, or what the acceptance criteria will be for that particular test.
Okay, so here's the last question that we'll get to today. What is the biggest challenge for reprocessing these types of devices?
Oh, that is a loaded question saved for just the end. Perfect. There's a lot of challenges. They are very intricate devices. They're very small. They oftentimes have small internal lumens so that the patient contacting robotic portions of the device often have small internal lumens and small internal mechanisms that can become contaminated and can be difficult to clean, and can be difficult to extract as well during the validation process. And so I would say, yeah, just the complexity of not only the end effectors of these robotic devices in some cases, but also the complexity of the internal components are those are going to be your biggest challenge to the validation process. And trying to effectively clean those areas as well as effectively extract those areas to show that the cleaning, or to demonstrate that the cleaning procedure is effective, that's going to be a challenge as well. So it's kind of two challenges: is one, can it be cleaned, but then also can we effectively extract those areas to show that it can be cleaned. Those are I would say your two greatest challenges, as well as kind of just simulating the use of the devices. Again, they're very complex devices that are going to be they're going to come in contact with blood, but also tissue. And so during the validation testing, you're going to want to choose an appropriate test soil, but also you're going to want to consider using tissue, as again, these devices are going to come in contact with tissue, and in some cases they're going to have cauterizing action, so you're going to want to take into account that process as well. And they're going to have cutting action and grasping action, so you're going to want to simulate that on tissue. And so I would say those are the greatest challenges. So again, just ensuring that you've adequately challenged it by selecting an appropriate test soil, as well as then just demonstrating that you can effectively clean the end effectors and those internal components, as well as extracting the end effectors and extracting those internal components during the validation testing. But yeah, if you want to discuss any more, if you have any other questions, don't hesitate to send an email to myself or to Mike, and we can definitely set up another call or have a conversation over email, and I'd be more than happy to answer any other questions that you may have.
Well, great. Thank you, Griffin, for this this discussion around robotic devices, and we appreciate your insights. We'd like to thank you, the viewer, for also attending this session. We hope that you found this to be a valuable experience. This session was recorded and will be available on the Nelson Labs website just within the next couple of days. Thank you, and we hope you have a great rest of your day.